好，第10條啦，就淨係睇 2P 啦。當我哋混合晒所有嘢啦，澱粉酶同澱粉撈埋咗之後啦，咁我哋咧都做咗唔同嘅 food test 嘅。咁啊，碘液測試啦、尿蛋白試紙啦，同埋呢個本納德測試。題目就問你啦，喺邊一個實驗咧，係喺實驗嘅開始同埋10分鐘之後嘅結果係一模一樣嘅？咁其實呢一條題目咧，就同2017年嗰條咧，可以話異曲同工啊。因為嗰年就問你啦，撈埋咗之後咧，一開波嘅反應，咁當年嘅答案咧就係 A 嚟嘅。一開波咧係唔會咁快有還原糖啊嘛，所以結果係陰性啦。咁但係啦，澱粉仍然喺度，而澱粉酶佢係蛋白質所做嘅，咁所以啦兩個咧都係正嘅。咁所以啦，基本2017年咧我哋知道啦，剛剛開波嘅話咧，消化應該係未開始嘅。10分鐘之後咧，就應該係有少少嘅 starch 咧係會俾人哋去拆解咗，而酶咧係用蛋白質嚟整嘅。咁今次個題目係問埋啊嘛，10分鐘之後又如何啊嘛？咁既然啦個反應已經發生咗，咁即係話有一部分嘅澱粉質咧應該已經係俾人拆解咗成為還原糖噶啦。所以啦 Benedict test 咧係一定有反應嘅。所以啦 Benedict test 就可以 say goodbye 啦，因為佢邊有可能一樣啫？由當初嘅 negative 就會變成而家嘅 positive 啦。咁尿蛋白試紙又如何啊？啲酶發生完反應係不變噶嘛，即係啲酶仍然響樹啦。咁2號仔咧係必然係一樣嘅，因為啲蛋白質仍然喺度。咁唯獨就係澱粉質啦。咁啊澱粉質10分鐘消唔消化得晒㗎？無論消唔消化得晒都好啦，一定係少咗澱粉質，呢件事係必然嘅。咁所以啦，我哋睇嘅結果唔係正定係負，而係個顏色嘅濃度啦。咁而家少咗澱粉質，所以佢嗰個藍黑色仲係咪咁深色咧？未必咁深色㗎啦，佢有機會淺色咗。所以碘液測試咧都係會唔同咗嘅，所以答案咧就只有 boy 啦。Question 10 we focus on the 2P. After mixing all the amylase and the starch, so we perform the food test, iodine test, Albert paper test, and the Benedict test. So we need to compare the result at the beginning and after 10 minutes. We'd like to find out which test will give the same result at the beginning and after 10 minutes. It's highly similar to the 2017 question. In that question, we are talking about at the beginning of the experiment. Negative results for the Benedict test and positive results for the iodine test and test of protein. So from the secret of 2017 question, we know that at the very beginning, the digestion has not yet taken place. And then after 10 minutes, a certain amount of starch will be broken down. And enzymes are protein in nature from 2017. We know something about it. That's why for this question, the critical concept, one of them will be enzyme remains unchanged after reaction. So that's why no matter at the beginning or after 10 minutes, the enzymes, they are still here. And enzymes are made up of protein. So that's why Albert test should give you the same result. So what about the Benedict test? At the very beginning, because there is no reaction at all, so no reducing sugar is produced. However, once the reaction starts, so the starch will be broken down into maltose. And maltose is a type of reducing sugar. So it will give you the positive result in Benedict test. So you can see that at the beginning, negative. After 10 minutes, there will be a positive result. So that's why for option 3, we can say goodbye to it. So what about the iodine test for the starch? So you may argue that, no, Mr. Lerm, the reaction may not be finished. It's no big deal. Once the reaction starts, the starch will be broken down into maltose by the amylase. So there will be less starch present. So for the iodine test, we do not only observe the positive or negative result. We can also observe the color intensity. So there should be a pale blue-black color. So that's why for the iodine test, it should also give you a different result. So that's why the answer will be B.